Bismillahirrahmanirrahim. I would like to welcome you to the fifth lecture on the series of lectures on research methods. Today we will be talking about some quantitative research methods. In today's lecture, we'll focus on the following things. We'll talk about different variables. We'll find out what do we mean when we say relationship between independent and dependent variables. We'll talk about some important types of variables. We'll also revisit hypothesis, samples and populations, and the concept of significance. Variables is a class of outcomes that can take one or more value. The more precisely a variable is measured, the more useful the measurement is. So, what is a variable? A variable is an outcome of a treatment. A variable is something that can take different values. For example, you must have studied this concept in your basic mathematics, that x is a variable and x can assume any value. So those values could be discrete numbers, or those values could be, for example, male, female, or those values could be level of education, high school, higher secondary school, college, university, or undergraduate, graduate, doctoral. These values will go in a variable. Now, what is dependent variable? The outcome of a research study is called a dependent variable, or the effect. It depends on the experimental treatment. Now remember, we are talking about quantitative research methods. So outcome of your experiment, the effect of your experiment is called dependent variable. Now what is independent variable then? Independent variable includes the treatments or the conditions under control of the researcher, the causes. Those are called independent variables because researcher can manipulate those variables, and there should be at least two different values of independent variable. If there are no two values available for that variable, how will you find out the difference? Whether the treatment had an effect or treatment did not have an effect. So there should be at least two levels of independent variable, or the treatment or the cause. Now what happens to the effect? If the effect is same with both the treatments or both levels of the independent variable, it means there was no change, there was no impact of independent change in the independent variable on the dependent variable. Relationship between independent variable and dependent variable. Independent variables should not be confounded. What this means is that levels do not vary systematically with other variables. If there is a change in other variable, in some other variable that is not under study for this research, level of this independent variable should not change. If it does, that is a confounded variable. And that means you do not have, as a researcher, you do not have complete control over independent variable. And if you do not have a complete control on independent variable, you cannot say anything for sure for the dependent variable. Dependent variable should be sensitive to changes in the independent variable. So we should have a pair of dependent and independent variable in such a way that the dependent variable should be sensitive to changes in independent variable. If dependent variable doesn't change to any change in the independent variable, it means that probably there is no relationship between the two variables. Well, that could be a finding in itself, but whenever we are selecting dependent and independent variable, we should keep these things in mind. There are other important types of variables. One type of variable is called control variable, and it has a potential influence on the dependent variable. So what do we do with this variable? We control this. We do not let control variable impact the values of dependent variable. That's why it's called control variable. The other important type of variable is called extraneous variable or intervening variable. Intervening variables have unpredictable impact on dependent variable, and they cannot be controlled. That's why they are called intervening variable or extraneous variable. So what do we do if we cannot control them? We need to infer their effect on the dependent variable. For example, if you are using working professionals in their actual professional environment for your research study, maybe for judging impact of using software agents in the work environment, there are a lot of variables that you cannot control. For example, someone might be sad or extraordinarily happy. So that will impact the results of your study. So that type of variable that you cannot control because you are doing your research in the actual work environment, you call them extraneous variable. Now you can make an attempt to control for it by using university students and make them behave like professionals. Now you might be able to control those variables, but you compromise on another thing. You will not get the actual results that you would in an actual work environment. Similarly, the next important type of variable is called moderator variable, and these variables are related to either independent variables or dependent variables, and they hide the true relationship between independent and dependent variables. So we need to take the effect of moderator variable into consideration before we finalize our study. Here's a summary of different variables, their impact and their definitions. Dependent variable, a variable that is measured to see whether the treatment or the manipulation of independent variable had an effect. This is called outcome variable, results variable, or criterion variable. Independent variable is the one that is manipulated to examine its impact on dependent variable. This is called treatment variable or factor variable or even predictor variable. Control variable is a variable that is related to dependent variable, the influence of which needs to be removed. It is called restricting variables. Extraneous or intervening variables are the ones that are related to either dependent or independent variables and are not part of the experiment. These are called threatening variables. Moderator variables are the ones that are related to either dependent or independent variables and have an impact on dependent variable. These are also called interacting variables. Hypothesis. We have already talked about this concept in one of our previous lectures. Here we will see how to scientifically write down your hypothesis. A hypothesis reflects general problem under study, restates the general problem in a form that is precise enough to allow for testing. If you remember, we called the hypothesis a tentative answer, an educated guess. And we said that hypothesis should always be written in a declarative form, which is testable. A hypothesis is a tentative assumption made in order to draw out or test its logical or empirical consequences. There's another definition: a belief based on an observation that has not been tested yet, or an educated guess. Now we will talk about different types of hypothesis. The first important type of hypothesis is called null hypothesis. And null hypothesis always assumes that there is no relationship between independent and dependent variable under the study. So what do we mean by that? That the mean theoretical average of population 1 is equal to the population theoretical average of population 2. And this is how we denote it. This is H naught, which means null hypothesis, colon mu1 is equal to mu2. What does this mean? This means that there is no difference between the theoretical averages of population 1 and population 2. And that's how we denote it. And we always start with the concept, with the notion that there is no difference and there is no relationship between dependent and independent variable. That's what we assume. That's the starting point for all research. Purpose of the null hypothesis is to provide a starting point for analysis, and it is accepted as true unless proved otherwise. And it assumes that chance may have caused any observed differences. When do you develop hypothesis? Because we have discussed this thing in our literature review section, that it might be based upon an observation. So you observed that if something happens, it has a different effect. And now when you want to actually test it, you assume that the observed difference was because of a chance, because something else had happened. So you want to remove that bias of your previous observation before you conduct this study. And it provides a benchmark for comparison. The second type of hypothesis is called research hypothesis. Research hypothesis is a statement of inequality as compared to null hypothesis. Null hypothesis was statement of equality, that there is no difference due to treatment. Now we assume that there is difference, and we describe that relationship exists between independent and dependent variable. And how do we denote it? Scientifically, we call it H1, and we put a colon. And now you see we are not using mu, rather we are using x1 bar. x1 is the theoretical average of sample 1, and we say that it is not equal to theoretical average of sample 2. We use an inequality symbol here. And what does this mean? This means that there is some relationship between independent and dependent variable. That's why when we were using those two levels of treatment, one theoretical average of one sample is different from the theoretical average of the other one, because there was some difference. Okay, now there is another interesting question that you might ask, that we used population in the previous hypothesis and we are using sample in here. That's because null hypothesis talks about the whole population. Whatever we do in our research, we want to generalize that research. If you remember from our first lecture, when we were talking about characteristics of good research, we said that good research is generalizable. And for example, if you want to find out behavior of Pakistani people, certain thing, you might not be able to actually go and ask every single Pakistani. We are about 160 million people. It is practically impossible to go to ask the same question to every single person. So what do? We ask that question to a sample, to few people, and based upon the responses from that sample, we generalize that result. So when we are doing our experiment, we always consider population. That's why we are using x1 bar and x2 bar instead of mu1 and mu2. Mu always stand for population. X bar stand for sample. Now there are different types of relationships between research hypotheses. One type of relationship is called non-directional research hypothesis. It means that two groups that you had are different, but there is no direction specified in the hypothesis. And how do you say that? That h1 colon x1 is not equal to x2. We do not know which one is greater or which one is lesser. This leads to the second type of research hypothesis, which is called directional research hypothesis. The groups are different and the direction is specified. For example, x1 bar is greater than x2 bar, or the other way around. So these are called directional and non-directional research hypothesis. What is the purpose of research hypothesis? Because they can be directly tested during a research process, and they provide us a place where we can compare them against null hypothesis. Here are some of the differences between null hypothesis and research hypothesis. Null hypothesis is a statement of equality between variables. Research hypothesis is inequality between variables. Null hypothesis always refers to population. Research hypothesis refers to sample. Null hypothesis is indirectly tested. Research hypothesis is directly tested. Null hypothesis is stated using Greek symbol mu, whereas research hypothesis is stated using Roman symbol x bar. Null hypothesis is implied. Research hypothesis is explicit. What makes a good hypothesis? We have talked about these things. That it should be stated in a declarative form, that it asks a question, it answers a question, and it posts relationship between variables, it reflects theory or literature, it should be brief and to the point, and it should be testable. Now we move on to the important concept of the difference between samples and population. A sample is a representative portion of a population, whereas population is the entire group of interest. And we have already discussed that results from samples should be generalizable to population. Significance means that what is the probability that your observed differences are from the treatment and not from chance. We need to find out the significance of your results because there could be influences of more than one treatment. You might be thinking that you are controlling your independent variable, but there might be impact of intervening variable or moderator variable that you cannot control. So in that type of situation, you need to find out, you need to tell the readers what is the significance of your research findings. And once again, what is significance? Significance is the probability that observed differences resulted from treatment and not from chance. Significance level is usually associated with risk, that you are not 100% certain that the null hypothesis is incorrect. So that risk assessment is very important. Okay, now what types of statistical treatments you can apply on the data that you collect as a result of this experiment, or a survey, or any other quantitative research method that you employ for your own research. There are two important types of analysis that you can perform. First type of analysis is called univariate analysis, in which you are interested in only one type of variable. And you might be interested in the impact of many variables on a dependent variable. Now we will talk about different types of statistical treatments that you can apply on the data that you have collected as a result of your research study. This research study could be experimental based, or it could be survey, or it could be any other type of quantitative research method in which you have got a lot of data. You might be interested in finding out the difference between an independent variable and dependent variable. What if you have more than one independent variables? You cannot use univariate analysis. In that case, you need to use multivariate analysis. What are the different things that we use in statistics? We calculate mean, we calculate mod, we calculate median, we do t-tests, we find out z-scores, we do regression. Regression is the projection that in future, if the trend in the past follows, this will be the future result. We want to find out correlations between different variables. We might even go on to structured equation modeling. And there are other different types of statistical treatments that can be applied to the data that we gather. In this lecture, we are not going into the details of those statistical treatments because they become part of statistics course. But we need to identify, we need to understand that these types of mechanisms exist that we can apply on our data that we gather as a result of the experiment or any other quantitative research method. So once again, why do we do all these things? We do all these things because we want to increase our understanding of how and why we behave and do the things we do. This is what's called research method. I look forward to seeing you in another episode of this lecture. Thank you very much. Allah Hafiz.